Hey, sweet. What's up? Team Harry Potter here. We are doing our video. Lost one of our group members, but that's okay. So let's go ahead and get started. So you welcome. You want to just do problem one because it's super easy.
Sure. ATCGAAGGTTA.
There you
Starting
go.
5 prime, ending 3 prime.
There you go. Boom. Money. Goal. Okay. So second problem. Oh crap! I just quickly. Let's see that. Let's see. Video. Okay, so the second problem says that it relates to question one. How does knowing the structure of DNA help you hypothesize a possible model for DNA replication? So basically, my answer was kind of along the lines that if you know the structure of DNA, that it's a double helix, then you can hypothesize how like it's copied in the double strand. Like if you see that like these double strands are circled around each other in it, then you could probably guess that like they're replicated off of one another. So that's what I said. What did you say?
I said like that they follow the ATGC rule and they're parallel to each other. So with that, we can figure out a model for replication.
Yeah. So, I mean, I kind of feel like that answer kind of has a lot of answers, It's like,
Yeah.
I don't know. What did the key say?
I don't know. Probably something along those lines I wrote.
Okay, let me look it up really quickly.
I think it said something about the ATGC rule.
Okay. Let's see here. Says the DNA nucleotides in the double-strand DNA must follow the ATGC base pair rules, and the two strands are anti-parallel. As a result, a possible model for DNA replication involves reading each template DNA strand in the 3' to 5' direction, producing a daughter strand DNA strand with the opposite plurality and a complementary DNA sequence that follows the ATGC rules. Yeah. Okay.
Okay, so what are 3 possible mechanisms of DNA replication? Describe how these 3 models differ from each other. So we have the conservative. That's an entire copy of the double helix is made. Semi-conservative, so like the 2 DNA strands separate and serve as templates for the synthesis of daughter strands. And dispersive, so the newly made DNA becomes dispersed in the original parental DNA molecule. After replication, the 2 daughter molecules each have 2 strands of DNA that contain both parental sequences of DNA and newly synthesized DNA.
Yep. Pretty much. Okay.
Wait.
Go ahead.
No, nothing.
Okay. All right. 4 reads the Meselson-Stahl experiment was based on the ability to separate heavy nitrogen, half heavy nitrogen, and light DNA nitrogen in an ultracentrifuge. Then it asks what would be the results after a single generation of each of the models. So for part A, after a single generation, the conservative would have half DNA with heavy nitrogen and half DNA with light nitrogen.
Right.
The semi-conservative would have 100% half heavy. So like a combination of the nitrogen 15 and the nitrogen 14.
Right, yep.
And same with the dispersive model. It would have 100% half heavy. Now, in part B, after 2 generations, the conservative would have 3/4 light and 1/4 heavy.
Wait, I'm just going to wrap my head around that.
Sure.
Yeah. Yep. Wait, so first they had 15, 14, and then it became half. Why is it 3/4, 1/4 of the heavy?
Because I believe that how the model was set up was that they created DNA that had nothing but heavy first, and then they put that DNA in a kind of like a solution, that has only the light, nucleotides in it. The
Yeah.
nucleotides with the light DNA. So when it replicates it, it gets the light DNA from the surrounding. So if it replicated again, since it's 50, it's 50% is heavy and 50% is light. The 50% of the heavy is going to divide. They're going to make another light, and then the light is going to make another light. So there's going to be 3 lights to 1 heavy.
But isn't there 50-50 of them though?
No, that would be three lights to 1 heavy. That's three fourths to 1/4.
Right. So there was. So in the first one we had. So we had. If it was on the conservative, we had 15 nitrogen, half of that, and 15 and then half 14 on nitrogen, right? After the first.
After the first, yeah, it should be 50-50.
So why isn't it 50-50 if they replicate again?
Well, because the light. So I always think of it like, let's start out that we had 1 heavy DNA, okay?
Mm-hmm.
And then after the first generation, that replicated to make 1 light chain, right? So that's why it's 50-50. So you have 1 heavy chain to 1 light chain, okay?
Wait. So wait, you don't start with what did you start with, you said?
You start with 1 heavy.
1 or 2 heavy, no?
Well, I mean, it doesn't matter, like, but here, hold on a second. Let's see if I can pull up the. Okay. Here's a good one. So I'm gonna do a screen share right here. Okay, so you can see my screen now. So if you look at these models right here, can you see my screen all right, Joachim?
Yeah.
Okay. So here you start. Here's the original double helix. You see that it's a heavy right here, right?
Yeah.
So when it replicates, according to the conservative model, it'll make 1 heavy right here and then 1 light. And the reason why it makes 1 light out of this 1 heavy is because this heavy stuff is in a solution of light.
Oh, okay.
So going back to this right here. So that way, anytime that this replicates, like anytime it's copied, it's going to make light. It's going to get light nitrogen in there. So again, in the second generation, this heavy one is going to, replicate another light one. And then this light one is going to replicate another light one. So there are 3 lights to 1 heavy.
Right, okay, I see that.
So,
and that's just because it's in the same solution, you said?
Yeah, it's because it's all in the same solution.
Okay.
So, and then furthermore, the semi-conservative, there would be half heavy and half light, or half
Heavy.
half heavy and then half light. Yeah, sorry.
Yeah. Yep.
And then in the dispersive model, there's still 100% half heavy. Yeah?
Right.
So, and when they actually ran the actual experiment, they found out that it followed the semi-conservative model. So that's the one that we adopt nowadays.
Mm-hmm. Yeah.
So, okay, does that make sense?
Yep.
Okay, cool.
So let's draw the DNA replication process. So draw a linear double-stranded DNA molecule. Indicate you're drawing the polarity of the parental strand and an origin of replication. So I just drew a 5' to 3' and 3' to 5' with like an origin of replication. I drew a circle around it.
Okay. Yeah, I drew it like this. You see it all right? So that top part right here.
Pick it up a little bit. Yeah, I drew a lot more simplified thing, but essentially the same thing.
Okay. Yeah.
So-
yeah. Let's see.
Oh wait, up here.
Oh yeah. Okay. Okay. Yeah, you drew the same thing as me. Yeah, you basically did. So.
Okay, so draw an early stage of replication. Indicate the two replication forks, 2 leading strands and 2 lagging strands.
My on that one, it's pretty similar to the one that I wrote.
Yeah.
I just wrote a couple of like nucleotides, and then I wrote like DNA polymerase, and I pointed to the nucleotides and said, "Hey, this is the Okizaka fragments."
Yeah,
I wrote
'cause
which ones were lagging too. So
I just kind of drew them the same way, and then like saying it's an Okizaki fragment in there.
yeah, that's basically what I did too. Like it was almost the same, except I added a couple of nucleotides in there being replicated.
Yeah.
And then part D says, what do the 2 new strands, like what's this a finished product look like? I mean, I just wrote 2 DNA strands.
Yeah, parental and daughter, daughter and parental.
Yeah. And like, yeah, and I wrote down their polarities, that they're anti-parallel, 5 prime, 3 prime, 3, 5 prime, 3 prime,
I never did that, but yeah. Okay, but.
Okay. Moving on to 6. So 6 reads, name and describe the functions of the enzyme in the 3 phases of prokaryotic DNA replication. Initiation, elimination, and termination. So part A says, which of the prokaryotic replication proteins consume ATP during catalysis? So the ones that I wrote were DNA gyrase, which is the
Helicase?
ATP to relieve positive supercoiling. It's topoisomerase.
Wait, so did you wrote? Oh, DNA ligase, you said?
I'm just taking each one. I did write DNA ligase, but I wrote others.
Which one was the first one you said?
DNA gyrase.
Is that the same as the helicase?
It's the same as the topoisomerase.
Oh, the 2?
Yeah, the topoisomerase too.
Okay, I got that one.
Yeah, so same thing, But yeah, that one uses ATP to relieve tension from positive supercoiling. I had DNA ligase, which uses ATP to link adjacent Okizaka fragments, those little tiny gaps, and it just puts them together. I put DNA primase, where it uses ATP to add a primer of RNA so you can begin replication off the lagging strand. And DNA helicase, which unwinds the DNA, breaks the hydrogen bonds. Is that all of the ones that use ATP? Did I miss any? Okay. On part B says, which of the prokaryotic replication proteins consume the dNTPs during catalysis? So I just wrote DNA polymerase 3 and DNA polymerase 1.
I just wrote DNA polymerase.
Okay. I think that works too. So, but yeah, what did she say in the key? So she says,
do you know like what all these stuff do? Like from memory?
yeah, more or less. Mm-hmm.
That's good. I kind of do, but I need to review it. And yeah, make sure I know them, because it seems like these are coming back.
He wrote DNA polymerase 3 and DNA polymerase 1.
Is that what the key said?
Yeah, that's what the key says. And also on part A, she wrote DNA A in there as well, which I was wondering whether to include that or not. I thought DNA A and DNA helicase might have been kind of the same thing, but I guess not.
DNA A?
Uh-huh.
How do you spell that? Like DNA, like a small a after?
Yeah, DNA and then a big A after.
So
it's like capital D, lowercase n, lowercase a, capital A.
Oh. Okay.
Yeah, kind of weird. Hey, FYI, I don't know why, but in part A of 6, You know how it says like list the ones that you consume ATP during catalysis?
Mm-hmm.
She wrote like every single enzyme in each phase, like initiation, elongation, and termination.
Oh, in 7, you mean?
No, in six.
Oh, wait,
So
what?
A, yeah.
During catalysis. Okay.
Yeah, she wrote like every single one, whether they consumed ATP or not.
Oh, yeah. Okay. Was it
Yeah.
a question though on the key?
The question says, oh, I see. Okay. Oh, so she want okay. Okay, I think I understand what's going on here. So
It's the same question.
yeah, I thought she just wanted us to answer A and B. I didn't know she wanted us to like. That's why I was kind of confused on this because
What did the key say though?
the key. So okay, so in six it says name and describe the functions of the enzymes involved in the three prokaryotic DNA replication.
Yeah, right. Yeah, so that one is not on ours.
Yeah, that was a question in and of itself, and I didn't
We have the
realize.
next one, which deals with eukaryotic DNA.
Yeah, seven. I did answer. You want to go ahead and answer seven?
Sure. So for the initiation, name and describe the functions of the enzymes involved in the three-phase of eukaryotic DNA replication: initiation, elongation, and termination. So for initiation, I wrote like the origin recognition complex. It binds to ARS at all times. What was ARS again?
Oh, it was. Oh man, I remember I had to kind of look through the notes to be able to find what the ARS was, but let me look it up again. Aha, here it is. Okay. The A. Yeah, the ARS is the autonomously replicating sequence.
Okay. So
It's basically the TATA box.
Oh yeah, that one.
Okay. Yeah, that's what it is. It's the TATA box.
So regular proteins was CDC6, CDT1 that binds to ORC. What was the ORC?
Oh, that's the origin recognition complex.
Okay, that was the thing we.
Just it's the thing you
just. Is it the origin of replication?
Yeah, origin of replication complex.
So it inhibits the initiation of replication, and then kinases phosphorylate CD6 and CD1, and in it like so that it initiates it. So first is apparently the CDC6 and CDT1 is blocking it, and then when kinases phosphorylate them, that's when it starts. And so like CD6, C6, and CDT1 kind of act like a blocker.
Yeah, pretty much. Like they regulate it, just as their name implies.
Then we have MCM, mini chromosome maintenance helicase. After CDC6 and CDT1 is released, MCM helicase catalyzes the separation of two parental DNA strands.
Yeah, basically it's helicase. It just separates the bands.
Right. So that's what I had for initiation. And then for elongation, DNA continues to be separated by the helicase. Then you have DNA polymerase alpha, replicates nuclear DNA, associates with DNA primate to form a complex that synthesizes RNA and DNA strand. That's used as a primer by DNA polymerase delta and elongate. What's that? E in Greek, whatever that is called,
Epsilon.
epsilon, right? For DNA synthesis. When RNA/DNA primer is made, DNA polymerase alpha is displaced and replaced by DNA polymerase delta or epsilon. Gosh, wait, what did I just say?
So epsilon.
Okay. Wait. I have more. So,
okay, more.
So DNA polymerase delta synthesizes the lagging strand and contain proofreading activity. Epsilon synthesizes the leading strand, also has proofreading activity. And alpha, what did that do? Replicates nuclear DNA, or what did you say?
Well, I didn't have the correct answer, but I thought alpha synthesized the leading strand, but I was wrong. That was epsilon. But according to the key says that so DNA primase adds the primer, the 10 nucleotides, and then it says DNA polymerase alpha synthesizes 20 to 30 nucleotides off the primer.
So wait, the primer does what?
So the primer comes down and it attaches to the template strand as kind of like a start point, if you will.
Right.
Then DNA polymerase alpha comes and synthesizes 20 to 30 nucleotides off that primer.
Right.
It's weird because DNA primase and DNA polymerase alpha are part of one complex. They're part of the same protein.
So are they just starting the elongation?
Pretty much, yeah.
And then delta and epsilon goes and continues, or what?
Yeah, and then delta and epsilon go and get the rest of it.
Okay. I also had tCNA, and then that says increases the processivity of DNA polymerase epsilon and delta. And DNA polymerase delta and epsilon also clamps pCNA to each template DNA strand.
Yeah, that's what I was about to say. I was about to say that it just kind of clamps them down onto it, so that way they don't.
What does that mean, though?
Well, I mean, you know how clamps work. They just hold them down onto it.
Oh, okay. See, I kind of have forgotten about this stuff a lot because I did it. I didn't do it this week. I did it last week in the beginning.
Oh, you did it last week. Okay.
So, this is way old. I'm what was all this stuff again? I actually haven't touched biology the whole week since I worked ahead.
Oh, gotcha. That was a smart thing to do. I should do something like that. I should
I still need to be ahead. I still have that week where I need to be gone a whole week. So now I've let myself be. So I still haven't done the next activity for this week. So I'm on target right now, or on time, I guess. But in termination, by the way, there's no there was no known proteins.
Do you know what our next exam is?
No. I do not. So yeah, that's it. I'll stop.